And so next, I'd like to invite Professor Chuang from the Dalian University of Technology. Where he's the Deputy Dean. And Professor Xue has got his PhD from the Dalian University of Technology and did a postdoc at the Ohio State University. And he's been at Dalian University of Technology since, and he's now a very productive scientist with more than 50 peer-reviewed articles, 20 patents, and many engineered strains that have been commercialized by companies. And he's also received many awards, such as the Gold Medal of the International Exhibition of Inventions of Geneva, and the first prize in the Scientific Technology Award of the China General Chamber of Commerce, amongst many others. So without further ado, I'd like to invite Prof. Xue to give us a presentation on the metabolic and process engineering for enhanced bioalcohol production. Yeah. Prof. Xue, please.
Okay. Thank you all, Beth and AUS, to organize this conference, and thank you, Chairman. I'm so sorry, Professor Chuang is doing another official oral defense at this time in China. So I'm here doing this presentation on behalf of Professor Chuang. Okay, I am Dr. Wu. I'm his team member. Okay, from School of Bioengineering, Dalian University of Technology in China. This presentation with the topic of metabolic and process engineering for enhanced bioalcohol production. Okay, this is the outline of presentation. The first part, background, less in two points, bioethanol as a second generation biofuel and attempts for improving bioethanol production. The second part, engineering kinesis in bioethanol producing stream, less in 3 points. Regulatory functions of histidine kinesis on solvent production or solventogenesis, morphological and transcriptional analysis, engineered strains with hyper-bioethanol producing capacity. Okay, the third part is membrane fabrication for bioethanol recovery. Okay, I will try my best to express the exact meaning and scientific finding of Professor Chuang. Okay, bioethanol is an important chemical with many applications in plastic and food industries. More important is also a potential biofuel. However, as far as we know, ethanol has been well commercialized for years. And ethanol fermentation is the largest industry in biofuel area. So for bioethanol to be a substitute for oil fuel, as bioethanol takes great advantage over ethanol. For example, bioethanol is higher energy density, has better compatibility with gasoline. Actually, around 10% ethanol could be well mixed with gasoline in tank. However, higher ethanol ratio will damage the car engine. For bioethanol, we don't worry about for this situation, because for 100% bioethanol in the tank, there is still no harm to the car engine. It's very, very safe. Okay, usually bioethanol could be produced by ABE fermentation. ABE named acetone, bioethanol, and ethanol, using Clostridia, such as Clostridia acetobutyllica. For now, the major challenges for ABE fermentation are low bioethanol titer, productivity, and high recovery cost. So, metabolic engineering and process engineering approaches provides possibility for enhancing ABE fermentation. The major solutions for case studies are summarized here in these years are stream engineering and fermentation optimization and memory fabrication for product recovery. Next, I will introduce some sporulation program in Australia. In the past 20 years, the sporulation program or clue in Australia as Metabolic has been well shown in somewhere. You see, first, Spor0A. Spor0A is an upstream, a global transcriptional regulator responsible for sporulation and solvent genesis. Okay, by knocking out or expressing this regulator Spor0A, however, the mutants didn't work well. They produced trace butanol without apparent solvent genesis. Okay, some other studies also focused on the downstream factor, sigma factors like sigma F, sigma E, and sigma G. They all involved in sporulation program. By knocking out these factors respectively or one by one, the mutant still failed to form any viable spore, but give normal butanol production. Is very interesting. Okay, that means the upstream sporulation related genes, Spor0H, 0A, and sigma K, can play big roles regulating butanol formation. While the sporulation behavior, the behavior itself is not very essential for butanol formation or solvent genesis. Later studies showed a good attempts for decoupling solvent genesis from sporulation, but still failed to further improve butanol productivity. Okay, how to well coordinate sporulation and solvent genesis remains a challenging task. For a long time, metabolic engineering of Australia remains a hot issue, a very hot issue. Various mutants here have been constructed by expressing or knocking out key genes of central carbon metabolic pathway. Yeah, various mutants here. Okay, although some preferred cellular traits or improvements were achieved here and there, these engineered strains still failed to produce more than 18 gram per liter butanol in batch culture. Also, engineering a stress response or transcriptional regulator gave limited improvements in butanol tolerance or titer. Actually, for these mutants or engineered strains, sporulation is somehow another problem for industrial fermentation. The spore initiation will cease or stop a solvent genesis and butanol formation earlier. So, non-spore forming and hyper-butanol producing Clostridia is more preferred in future. However, conventional metabolic pathway engineering approaches couldn't affect or eliminate sporulation program. Based on this, histamine kinases may be a new key target for improving AB fermentation in terms of their regulatory functions like on-off switch to sporulation program. Okay, as reported, Spor0A is a major solvent genesis regulator where phosphorylation or dephosphorylation by histamine kinases. A different histamine kinases also show distinctive regulations on sporulation program or frequency. For example, here is for histamine kinases encoding gene, or encoded by gene CC3319, CC0323, CC0903, and CC0437, or these 4 genes. By knocking out, disrupting this single gene, okay, 3 mutants here, 1, 2, 3. 3 mutants showed decreased sporulation frequency from 1% to 5%, while 1 mutant, the last one, showed increased spirulation frequency up to 34 times. So all these lice spore forming mutants provide important target or clue for regulation, sporulation, or thermogenesis. In a word, we attempt for not only decoupling thermogenesis from sporulation, but also improving butanol titer and productivity. Okay, this is part one, synergistic engineering of histone kinases for improving butanol production. Here, I will try my best to explain how single or multiple kinases regulate cells at global levels. Okay, the major limitation of conventional genetic tools lies in multiple genes engineering at one time in a round. Okay, taking a file histone kinase genes, for example, here 1, 2, 3, 4. A single gene was knocked out at each time. So one by one gene knocking out requires file rounds of transformation, screening, and resistant marker deletion. Okay, it takes so much time with low efficiency. So constructing a mutant library is very, very necessary, essential, but a tough work. So we here successfully developed such a method named multi-plasmid co-transformation for generating our theoretical mutants in one round at one time. Okay, we design inserting site of target gene and optimize the recovery time of transformant. And at least optimized genome editing method can generate our theoretical mutants, including a single, a double, a triple, a quadruple, and quintuple, knocking out combinations. For 5 genes knocking out, it usually takes 242 days by conventional strategy. Now it takes only 11 days. Okay, in our study, there were 5 target histone kinase genes. Here, 5 genes. In theory, 31 histone kinase mutants can be constructed by knocking out these 5 genes in different combinations, a single, double, triple, and finally the quintuple knockout. Okay, using the multi-plasmid co-transformation method above, we finally and successfully generated mutants library containing 22 engineered strains with single, double, triple, quintuple, quadruple genes knockout. Quintuple knockout transformants is not available. That means a total of 31. Now 22 engineered strains obtained. The other nine transformants of genes knocking out combinations cannot survive well. Among these 22 engineered strains, knocking out a single gene, CAC3319 showed the best fermentation performance and playing the most important roles for cell survival and thermogenesis. Okay, for each histone kinase encoding gene, for each gene, there were 3 engineered strains, including gene knockout, a gene complementation, and a gene co-expression. As can be seen in this picture, PS means a parent strain. A KO means gene knockout in parent strain. KOPMP1 means gene complementation in knockout strain. PSPMP1 means gene overexpression in parent strain. Okay, we then evaluate the effects of different gene expression levels on cell growth, butanol formation, and acid production accumulation during the whole AB fermentation process. Okay, gene knocking out strains or mutant could be constructed by designing and inserting special intron fragment into specific sites of targeted gene. These sites could be also optimized and located somewhere upstream or downstream of the targeted gene. In the following work, the relegations of gene CC3319 and CC0437, these two genes, were further verified by designing gene disrupting or intron inserting sites. For example, we constructed a CC3319 mutant with intron inserting site at the DHP domain. That means the gene function was severely damaged. Finally, eliminating the phosphor transfer activity of CAC3319 created a hyper-butanol producer and go a high butanol titer up to 17.5 gram per liter. Also, we constructed another CC0437 mutant with intron inserting site at the PAS domain. This mutant produced only 6.5 gram per liter butanol and more than 18 gram per liter acids above products in the fermentation process. As for the morphological changes of CC3319 and CC0437, knocking out mutants compared to control pattern apparent strain, the CC3319 knocking out mutant showed negative forms here, negative forms throughout of AB fermentation, different phases. These mutant cells were active in savantogenesis, acidogenesis, and decline phase. On the contrary, the CC0437 mutant showed similar morphological changes with the control parent strain, especially at the decline phase. A cell autolysis occurred. The wild-type strain ATCC55025 used in this study is non-spore forming Clostridia. It's natural. Its sporulation program is blocked. The wild-type sporulation program is blocked between stage 3 and 4. So the wild-type strain could form endospore, but couldn't generate a mature spore. Also, as can be seen here, knocking out the gene CC3319 blocked sporulation process at early stage 0, and kept cells at a wild-type form as a main butanol producer. Transcriptional analysis showed three genes related to cell autolysis: CC0554, CC1108, and CC1876. These three genes related to cell autolysis were all repressed in the CC3319 mutant, but upregulated or activated in CC0437 mutant, thus leading to different morphological changes. Okay, this is transcriptional analysis of central carbon metabolism related genes in these two mutants. Okay, as a CC3319 mutant give a high butanol titer up to 18 gram per liter, while another mutant CC0437 mutant produced only 6 gram per liter butanol and more than 18 gram per liter acids as byproducts. There must be changes in osteogenesis and thanatogenesis genes expression profiles. So at 4, the CAC3319 mutant, its sole operin was upregulated by 4.1-fold. This operin contained genes ADHE1, CTFA, and CTFB. They regulate protein formation and acid uptake. The acids, for example, acetate and butyrate, will uptake for butynyl formation again. So, upregulation of sole operin in the CAC3319 mutant enhanced butynyl production and decreased acid accumulation. As for the CAC0437 mutant, on the contrary, another mutant, the sole operin showed 10-fold downregulation. So more central carbon flux was towards acids rather than butynyl. Actually, butynyl titer is highly related to butynyl tolerance in fermentation broths. Okay, in our study, the CAC3319 knocking out mutant displayed better cell growth and higher cell density and improved butynyl tolerance and butynyl challenging conditions. Here, 5 gram per liter and 10 gram per liter. This is because the heat shock genes like DNAK, DNAJ, and HSP18 were upregulated, especially for the gene HSP18, 7.4-fold changes compared to its parent strain ADCC55025. On the contrary, CAC0437 knocking out strain mutant showed the blue one, shows a weak growth, decreased butynyl tolerance, and titer. Its shock genes like DNAK, DNAJ were all downregulated. The wild-type strain ADCC55025 parent strain is non-spore forming and so much preferred in continuous fermentation without sporulation. Okay, in the following work, we performed repeated batch fermentation with FBB to see their industrial potential. Here, the wild-type strain ADCC55025 and is hyper butynyl producing mutant by knocking out gene CAC3319 and CAC0323 double knockout were immobilized with MPB during repeated batch fermentation. The wild-type strain produced 13 gram per liter butynyl with lower yield and productivity due to its prolonged lag phase. The double knockout mutant produced higher butynyl titer up to 19 to 21 gram per liter with doubled productivity in 4 cycles. Okay, knocking out the gene CAC3319, as long as you knock out this gene, the cells capture vacuole form with faster growth rate and earlier thanatogenesis initiation. So this double knockout mutant here, strain double knockout, would have a great potential for industrial application. So according to these results about histone kinesis showed a global regulations on cell differentiation, essential carbon metabolism, and stress tolerance, especially for gene CAC3319 and gene CAC0437. They both responsible for SPO0A phosphorylation, for example. Knocking out the gene CAC3319 will enhance cover sensing and butanol formation, but repress sporulation process and acetate formation. Here, we also develop a high-throughput cloning and screening platform for yeast and Clostridia for industrial applications based on the whole genome network models. Gene editing was first performed to regulate cellular energy mass development at global level. A high-throughput molecular cloning and microbial screening system were then used for new strain cloning and screening with preferred cellular traits or improvements. Okay, especially this platform includes incubator, centrifuge, plate washer, online PCR module, microbial clone picker, and microplate reader. Okay, in part 2, we develop a high-performance membrane for butanol recovery to well-coordinate trade-off between permeability and the selectivity of butanol. We fabricated aligned carbon nanotube membrane. This is a fabrication process aligned CNT PDMS membrane. First, we use chemical vapor deposition method, making carbon nanotube to grow on silicon matrix for aligned CNT. Second, we use argon plasma to etch the CNT, making the tips open. Third, the PDMS polymer was being coated into aligned CNT. Then, hydrofluoric acid was used to make silicon matrix peel off. Next, the membrane was overturned. Then, the argon and carbon tetrafluoride plasma were used to treat the membrane. Finally, both CNT tips were open and added with PDMS layer, providing a nano channel for butanol or production carry. This is characterization of CNT arrays. It can be seen from the ICM and TM images compared to the untreated CNT. The treated CNT was highly aligned and a hollow tubular. Plasma remove metal particles and hollow core and open the tips of CNT. The inner diameter of CNT is 9.6 nanometer and 30-fold higher than the diameter of butanol and water molecules. Okay, this is a characterization of aligned CNT PDMS membrane. It can be seen from the TM image about CNT orderly distribution in membrane. The catching rate of argon and the carbon tetrafluoride plasma to PDMS is faster than that of CNT. The surface of PDMS is hydrophobic. Here, for butanol separation by evaporative membrane, it usually follows 3-step solution diffusion mechanism. The rate limiting step is a molecule diffusion through the membrane. Okay, in the PDMS matrix, the cavities for penetrant molecules are usually several angstroms in size. So the penetrant molecules must jump from one angstrom cavity to another. Finally, diffusion to the other side of the membrane. In this study, the open-ended CNT provide a channel for butanol diffusion with 15% flux increase and the butanol separation factor more than 100 achieved. Oh, okay. These are density functional theory calculations. Here, this showed penetrant molecules will pass through the CNT internal channels, not the PDMS chains or the CNT external wall. The order of absorption energy of molecules onto PDMS chain and CNT is as follows: first butanol, then ethanol, and lastly water. The CNT has the biggest absorption energy to butanol, and the butanol has priority to pass through CNT. The aligned CNT membrane breaks the trade-off between permeability and selectivity of butanol with increased flux and butanol titer. Okay, finally, we use a double gene CC3319 and CC0323 knockout mutant, double knockout mutant for AB fermentation. This gene can keep vegetative form throughout fermentation process without sporulation. Okay, it's non-spore forming and hyperbutanol producing. So the long-term culture can continuously produce butanol. When using the fabricated CNT membrane to remove butanol from fermentation broths, higher and highest butanol production can be easily achieved. So our integrated fermentation with product of butanol recovery system can reduce energy consumption to 2.4 megajoule per kilogram, so much lower than conventional distillation method, 36 megajoule per kilogram. Okay. In the conclusion, first, histone kinases have synergistic but distinctive functions on enhancing stress tolerance, cytogenesis by regulating cell differentiation and sporulation program. Second, the cell immobilization can stimulate the histone kinase engineer string to produce more butanol, up to 20.6 grams per liter, with high productivity of 0.46 grams per liter per hour, with high cell density. Third, the aligned CNT membrane breaks the trade-off between permeability and selectivity of butanol, with increased flux and butanol titer. In a word, the microbial butanol production or AB fermentation could be enhanced where this combined metabolic engineering and process engineering strategies. Until now, our research group have published more than 15 peer review papers. Among these papers are 3 ESI highly cited papers. SI has more than 2,000. In general, these publications include topical screen development and theme engineering, process engineering and product recovery, green biorefinery and waste action. Also, we have applied for more than 10 Chinese patents, 1 US patent. Especially, we have developed a full use and zero emission program by converting agricultural residues into valued biofuels of fertilizer, biofuels such as ethanol, butanol, and methane. This program and related technology were awarded with a gold medal in the 48th Geneva International Exhibition of Inventions. Here, many thanks for the support from the Ministry of Science and Technology of China, the National Natural Science Foundation of China, and many thanks for the help from Professor Yang Shangtian, Jiang Weihong, and Gu Yang. Okay, that's all for this presentation. On behalf Professor Chuang, many thanks for BAS and NUS to organize this conference. Many thanks for the chairman, and so sorry for missing this chance to communicate with all the friends here. I'm Dr. Wu, because my research area is waste biomass pretreatment, hydrolysis, and fermentation. So somehow my work fails to exactly express the meaning of founding the Professor Chuang. Still, thanks a lot for your consideration. And finally, by the way, globally, we'd like to recruit professor, associate professor, and postdoc here. Welcome to Dalian University of Technology. Thank you all. Thank you all.
Okay, thank you, Dr. Wu. Any questions? Okay, there is one question on whether you can elaborate a little on the CNT membrane separation of butanol. Is it used together in the reactor, in the fermentation reactor? And how do you avoid cross-strigure growing on the membrane surface if it's used together?
Okay, Professor He, I know you published several papers about maybe fermentation, but I'm sorry.
Thank you.
I did little work about CNT membrane fabrication. I'm so sorry.
Okay, I guess it's...
Yeah.
Go ahead. Please go ahead.
Professor Chuang fabricated this membrane.
Yeah, I guess, yeah, Professor Xue's email is right on this slide. So if you anyone has any questions regarding his strains and membrane technology, you can email him and get further clarifications or for collaborations. Okay. And another question is, currently, is this butanol in industrial manufacturing phase or not?
No, not the industrial, not the industrial.
Okay. And, yeah, do you know what scale they are doing it at?
It's hard to say, at least not the industrial.
Okay, that's okay. So
okay, I'll stop sharing.
Yeah. Okay. With that, I'd like to thank Dr. Wu for doing an excellent job standing in for Dr. Xue.